Hi students, in this video we will discuss about another pattern of inheritance called pleiotropy. Now what is pleiotropy? Normally a gene controls one character. In pleiotropy it is noticed that a single gene can control multiple traits. So that is what is known as pleiotropy. So a pattern of inheritance in which a single gene controls multiple traits. The gene is known as pleiotropic gene. So here you can see a chromosome and the location of a gene. This gene is a pleiotropic gene. And this gene is capable of controlling multiple traits such as trait A, trait B and trait C. So that is what is known as pleiotropy. Now some examples of pleiotropy. A gene B, that is a pleiotropic gene. This gene controls two traits. One is starch synthesis and the other is seed shape. And this gene in the homozygous dominant condition capital B, large starch grains are produced. And in the homozygous recessive condition small b, smaller starch grains are produced. Whereas in the heterozygous condition, capital B, small b, intermediate size starch grains are produced. And it is seen that the same gene B controls seed shape. The gene in the homozygous dominant condition and heterozygous dominant condition produce round seeds, whereas homozygous recessive condition wrinkled seeds are produced. This B is a dominant gene. Now if you notice the example of starch synthesis, it comes under incomplete dominance too. Because homozygous capital B produce large starch grains, homozygous small b produce smaller starch grains and heterozygous capital B, small b produce intermediate size starch grains and not large starch grains. So this comes under incomplete dominance. Another example of pleiotropy is phenylketonuria. This is a genetic disorder and it comes under inborn error of metabolism. Inborn error of metabolism means a child born with this disorder. It is seen at the time of birth, though because it is a genetic disorder. So normally what happens in our body is one amino acid, phenylalanine, gets converted to tyrosine in the presence of an enzyme, phenylalanine hydroxylase. So that is normally taking place in our body. One amino acid, phenylalanine, gets converted to another amino acid, tyrosine, in the presence of the enzyme, phenylalanine hydroxylase. But in this disorder, due to a single gene mutation, the enzyme is not synthesized. As a result, phenylalanine do not get converted to tyrosine. Instead, it gets accumulated in our body and gets converted to pyruvic acid or phenylpyruvic acid derivatives, which damages the brain and cause mental retardation and reduction in hair and skin pigmentation. So one gene is responsible for the disorder phenylketonuria and also it causes mental retardation and skin pigmentation. So it comes under pleiotropy. Now the next topic which you have to learn is chromosomal theory of inheritance. Before coming to that topic, you should understand that Mendel published his work in the year 1865. But this work remained unrecognized till 1900. That is, for 35 years, Mendel's work remained unrecognized. Now why, or give reason why Mendel's work published in 1865 remained unrecognized till 1900. The reasons are, all of you know, during that period, communication was not that easy and his work could not be publicized. Another reason is, Mendel stated that the factors are discrete and stable. But his contemporaries believed blending inheritance and hence it was not accepted. Also, Mendel's approach of using mathematics to explain biological phenomenon was totally new and unacceptable to many of the biologists at that time. And also, Mendel's factors or genes, he could not provide any physical proof for the existence of factors. So these are the reasons why Mendel's work remained unrecognized till 1900. One is communication was not easy and his work could not be publicized. Another reason is the concept of genes as stable and discrete units and the pair of alleles did not blend with each other was not accepted by his contemporaries. Also, his approach of using mathematics to explain biological phenomena were not accepted by the biologists of his time. And there was no physical proof for factors or genes. So for these reasons, Mendel's work remained unrecognized till 1900. Now the rediscoverer of Mendel's work are De Vries, Correns, Schermak in 1900. In 1900, three scientists, De Vries, Correns and Schermak, independently rediscovered Mendel's results on the inheritance of characters. And the chromosomal theory of inheritance was put forward in the year 1902. So that you will be learning in the next class. So I hope all of you have enjoyed the topic. If so, please click subscribe and also for the detailed notes, visit my website www.ranipradeep.com.